So I would certainly like to thank Asim Ansari and the other organizers of this meeting, which is really a spectacular meeting, for inviting me to participate. I came to town in 1968 doing a postdoc with Václav Šabalský, and my recollections of Corona, I'm going to just share a couple of things that just particularly I know about. Perhaps some other people have shared a few of these things, but the it's like I got to turn to the left here to get the mic working. So I had the experience of arriving not as an assistant professor, but just basically a recent graduate from graduate school, and I got ensconced in Cybulski's laboratory, and I got a phone call, and here it was, Corona. Wanted me to come over and talk with him and participate in some of his graduate or his group meetings with Raj Bhandari and other members of his group. And I must say it was one of the nicest experiences to kind of be welcomed, even though I'm not sure I was totally invisible, as was mentioned by one of the professors at MIT, but he had a very good way of making people feel welcome, drawing them out, and I didn't spend a lot of time in his lab, but 4 or 5 times we had conversations on some pretty deep science, and I also took advantage of when he finished synthesizing the tRNA gene. I used it quite a bit as a selection marker. It was an amber tRNA. The last time I saw him for this meeting, we were at a Gordon conference, a nucleic acid Gordon conference, and I had a car, and I drove him back to Cambridge, and we had another delightful conversation over a couple hours while we were driving, and then he invited me over to his house for dinner, where I met his wife and so on, which brings me to my third anecdote, and this came from Van Potter, who was actually the scientist or person here at Wisconsin who, shall we say, discovered Corona at British Columbia and brought him here. And one of the stories that Potter told me was when he went to visit Corona at Vancouver, he found out that, of course, he wasn't very funded. At that time, he was trying to synthesize the first nucleic acid, which was just poly-T, and his fraction collector for this purification was his wife, who came in every whatever amount of time was needed between the fractions, going all night and collected the fractions. So that really makes Faraday look like a piker, I think, in terms of finish. The final thing that I remember was a story by Günter Blobel, whom I saw in Germany not more than a couple of years ago, and he was in Madison at the same time, and actually I guess was in Potter's lab, but at least the postdocs and graduate students would socialize. But what he told me that was an interesting story was one night they were going somewhere, and then at the middle of the night they had to go back to the Enzyme Institute to tend to some experiments. It probably was some other postdoc in Gobin's lab that led them headed across the parking lot into the Enzyme Institute. And all of a sudden, there's tear gas, there's police with approaching them, accosting them, probably similar to the Gates situation that went on in Cambridge, but worse, as they had tear gas. But what it sort of reminded me of was that Madison at that time was a battleground between the police and the students in many ways. There was a lot of unrest, basically focused on the Vietnam War, but also focused on the relatively reactionary Bill Dyke administration. And you know, I'm sure Corona, just this sort of went past him. But certainly when I was listening to Al Cacharo's talk on the ethics and some of the ideas in which just getting the right policy is enough to solve problems when there's different parts of society that believe different things. But what actually stopped the unrest in Madison was an explosion in which a grad student named Robert Fosnack was killed. And the reaction of the town was very rapid and was very quiet from that point forward. The legislature passed a doubling of out-of-state tuition, and that about did it as far as they were concerned. So I think it's Goban had a very, probably, well, very important part on my scientific life, but a small part, but an important one in sort of framing my thinking about science. Anyway, I thank Goban for his existence. I've got 4 points in this talk, and the first one is the introduction. As someone mentioned, I was involved with, or perhaps more than just passively involved with, trying to sequence the genome of E. coli. At some other meeting, Ham and I could possibly exchange reminiscences about the sequencing of the first genomes. The only thing I will say, and Ham beat us roundly, but I will say our genome was quite a bit bigger. And we had sequenced more of E. coli and base pairs than the Haemophilus genome was. But nevertheless, this was a very big project, and it was a very important thing to me to begin really the idea of understanding a complete life form from a systems biology point of view. My idea of systems biology had to do with finding out the function of all the genes first and then seeing how they might work. This is a slide in which I just give some very summary information about the E. coli project. MG1655 was used. It was, for some reason, that I can't see, I don't know if you can, 4 million base pairs long. And the little sunburst diagram in the middle of that photo, or that illustration, there's some lighter yellow stuff that sticks out further, and that is the stuff that really seemed to be out of place. It was unusual in codon usage and unusual in some other properties, and it was kind of the first hint that there was a kind of a chimerism going on here. And that turns out to be, I think, the most important, or one of the most important things about the discovery. And at the moment, there is a fairly good percentage of the genes whose function is now understood. I think there's still about 12% that are more or less of unknown function. But the way you can look at the genome now is to say there's about a 20% genetic load. A genetic load being stuff in the genome that is, maybe you'd call it deleterious, but that's a point of view thing as to what the genome is there for. But I decided to adopt one concept, that we were going to try to use the genome in laboratory and production settings, so at least in terms of that goal. But another thing that was a very surprising aspect was that I think all the members of the genetics department that I was in, and I fitted in sort of as any physicist, biophysicist guy coming into a classical genetics department would fit in, full of very brilliant people. I was able to talk with people like Jim Crow and Julius Adler and begin to get some understanding of both the complexity of biology, but terms like genetic load, which are not at all obvious, and I'm not sure I understand it. But I would disagree with Mark Potashny in one way, in saying that everything is just an add-on to make it work better. It is more my view that the genome of, and there's more than just E. coli, is a bit of a muddle. in which different things are possibly added on, subtracted, thrown in. They're doing things that they just do. Not that a muddle is an all bad thing, but I don't think it's quite as simple as just a forward progression of either evolution or function. I'm gonna go a little bit faster now, but I wanted to get a little bit of that philosophy out. Probably 100 more E. coli strains have been sequenced. The rate of sequencing of those things is going up dramatically. Many are pathogens, a few are other lab strains. We've done one that is important for protein expression, BL21, and we've done a couple that are used for DNA expression in practice. And what you find is this pattern, of course, of horizontal transfer, mixed up genomes, includes IS sequences, which are basically the elements that can transpose. Prophage are pieces of or entire bacteriophage viruses in the genome. Sequences are genes with pathogen potential. No, that's a misspelling. I was just trying to cover it up. And and et cetera. And I know more about the et cetera, but I'm not going to make this a long talk. So you can do these Venn diagrams. If you only have 3 comparisons, they come out fairly horizontal or planar. And we did 3 different genomes in relatively short order for the time. A enterohemorrhagic bacteria, O157H7, which is up in the upper left, K12, the lab strains on the right, and a uropathogen, CFT073. And the central 3.7 megabases is basically what we call the core genome, and we coined the term pathosphere for all this up to 20% of genome material that is different or unique to the strains. And in this diagram, the area inside the Venn is a 7.13 kilobases, and the percentages are of that total, or of the pathosphere. And later, the term pangenome was published as indicate, and a valid thing to change the focus because it's really quite clear that all of the things that are in this universe of genes that can come in and out are not related to pathogens. But it goes to negate another of Alta-Charo's points that the first person who puts pen to paper doesn't necessarily win, because the pathosphere is a term that has definitely fallen out of favor, even though we discovered the concept of it. Anyway, the BL21DE3, one that I'm interested in, has more IS sequences, more phages. It has a couple of other things, but it fits exactly the same pattern. It's collinear, even though it's been separated in evolution as far as the main genes. There's a lot of mysteries here. But this whole thing inspired us to decide on a kind of another lifetime project of trying to study the genome by removing things from it and seeing if some principles and some increased utility could develop. I know there is a idea that finding the minimal or smallest genome is say even under a certain condition like a minimal medium is a goal, but it really isn't possible or even realistic to think of just reducing it as being all that informative, because of the basic point that I would like to make, that just as the genome is a mishmash, probably accidental in many ways, enabled by horizontal means by which DNA can just jump around from place to place, from genome to genome, but the environment is also a mishmash, and so what's minimal for one guy is not going to be minimal in another case. So we decided to just call it reducing, and we start in the upper left with the full genome and removed very large pieces that could contain as many as 80 genes down to some smaller pieces that could include just one gene, basically trying to remove genes that we either thought were unnecessary or didn't even know what they did. And what the company is interested in doing, Scarab Genomics, is trying to find strains like they're emanating there from MDS42 that can be used to make DNA or RNA or protein or low molecular weight compounds like biofuels or amino acids, proteins that are either periplasmic or cytoplasmic, RNA, maybe RNA interference compounds, and so forth. Then in the more speculative or whole genome or whole organism direction, up in the right, probiotics, vaccine deliveries, antibodies made through a reduced periplasmic strain. So the scheme is both a research project and an attempt to develop some useful tools that can be used to in commerce. Nomenclature, multiple deletion strain is what we call these. We name them MDS with an X. And then X stands for the cumulative number of deleted segments in the backbone. And we're basically going down a backbone structure in which the strain is not compromised. We don't remove either essential genes or conditional genes or genes that would have useful functions. Those get altered in the radiation to make the individual practical strain. So the number of genes is a lot bigger than the number in the nomenclature. MDS0 is the base strain MG1655, which is the K12 prototype. And when there's a T in front of the number, it means it's temporary designation. That's a lab designation. And the reason for that is after, the way this is being done is we really take the base strain and make the deletions one at a time and do enough characterization to see if, at least in isolation, it has any negative effects or any other effects on the behavior. And then we combine it in with the rest of the growing backbone sequentially and then check again to see if anything happens due to interactions. We have switched from an earlier stage when we used the wild type as the starting strain, then we moved up to MDS12, and now we're using MDS42 for the base strain for making the deletions, and we're going to shift up to MDS69 in a while. And we get these diagrams showing what's been deleted. This has got more deleted. And here is a graph of the rate of growth on the doubling time of MDS series on MOPS minimal medium. It's a balanced medium that Neidhart invented that sort of runs out of everything at the same time when it goes into stationary phase, so that anything that interferes with sort of any significant aspect of metabolism will probably slow down the growth. And for quite a ways, there was little bits of ups and downs in the doubling time, but not really a very big trend until we got up to about MDS 59, 60 in that area. And then between 62 and 63, the speed of growth of this thing really increased quite a lot. And our track record right now is 49 minutes from a starting number of 60, which sort of went up to 72. I don't want to vouch too much for the accuracy or the the precision with which different strains grow, but it sort of is an interesting thing, and we don't understand why, that at a certain point the strain started growing faster. Now we get to some micrographs. Basically, these strains look about the same size as a regular E. coli with the same morphology. They don't seem to have any real stringiness to speak of. here we just show that they basically segregate their chromosomes and sort of look like any other E. coli, look like a good healthy strain. Different ones are listed on the left. Since it's cut off, we got the base strain, MDS42 and MDS67 showed here in the horizontal things, DNA and RNA, and so forth. This is a dye that gets into dead cells, or at least membrane permeable cells. And it's interesting that MDS69 has a very strong viability. These are, I don't know what's going on to make the magnification take off the thing, but MDS42 is really quite viable. Now that's better. So MDS66 and T70 show a little bit higher proportion of dead cells. T69 is exceptionally good, and it looks like it's going to be our next main line. Now these are atomic force microscopy strains and pictures in which the MDS42 strain, which has basically all the fimbrial genes, all the flagella genes removed. You have to look a little bit hard to see the, do we have one of these pointers? You can see the flagella hanging out here, and you can see the fimbrial hair-like structures here. These are basically bald. And between 42 and 67, we removed quite a lot of periplasmic genes, and there is what happens when the periplasm gets emptied. So, and this just shows the reduction in the periplasmic strain. This shows the ability to grow anaerobically, aerobically on xylose and glucose for a couple of the strains. Reduction in biofilm production. And this is a list of the properties. And this is a list of how these properties might improve the performance by reducing energy wastage, just simply by eliminating the things that need to be made, stabilizing the genome by reducing mutation rates and eliminating transposition, and so forth. Now, point one is that IS hopping transposition can itself be induced by the stress of plasmid growth and protein expression. This is any old plasmid growth that you buy or make with any strain. The only strain that doesn't have IS elements in the plasmid preps comes from the reduced genome strains, such as shown there, MDS42. Here's an example which we have published in which the IS transposition was so great that it actually prevented getting a clone of this protein expression line. Different ISs going into different places. These gels just simply show the sizes of the plasmid by restriction mapping. The only way to get this thing to grow was to put it into an MDS strain. This shows something from the GenBank database where a whole piece of the DH10B chromosome was introduced into a back by just simple transposition by IS10, which is a remnant in DH10B from a previous genetic construction done in the lab, which wasn't known to have remained. And for point two, yep, I'm over already. Oh, I didn't get a prior warning. Well, I think I will, I'm well over time, but I'm going to try to finish the points I'd like to make, if it's okay. The same thing is true for the stress of protein production. And I'm going to show you an example here in which this simply shows we can make the protein in fed batch culture. But comparing this with the commonly used strain under the same induction protocols, the yield is quite a bit higher for the reduced genome strain, around 10 grams per liter versus about 2. And the reason is because the production strain basically lyses about halfway through, because the stress of the overproduction of the protein basically induces the prophages that are making lysozyme that lyses the product, which is shown on the right there by looking at the Western blots of the cell-free supernatant of the production fermentation compared to the reduced genome production strain. Now, this can actually be illustrated by means of another approach to making an alteration of the genome, which was actually done at the inspiration of Max Gottesman, who has a antibiotic that interferes with the Rho factor, and he discovered that 2 termination factors could actually be deleted from the reduced genome strain, whereas they were essential in the wild type. So we just decided to see if the drug that inhibits the Rho factor has any effect on the strains. And as you can see, the wild type strain, the bicyclomycin inhibitor of termination causes lysis, whereas in the MDS strain, it doesn't have an effect. And that has to do with the fact that that one of the adaptations of E. coli is to have termination sites that are existing outside of prophages so that they can't read it through into the genome. And on the other hand, the phages have developed anti-termination factors in order to get past that defense, which kind of illustrates there's a little structure to the mishmash. So the conclusion that I really want to make, and since we're working in aphorisms in this meeting, this one came from Click and Clack. Somebody sent in a letter asking if it's possible for 2 people to know less than 1. And I think the answer to this is yes. But it reminds me that if you have 2 or more genomes in a cell, that can also be dumber than 1. And I think the genome of E. coli contains many elements that are at cross purposes, which may partly explain why rather unstructured, again I'm misspelling, genome reduction can bring about really substantial improvements. Now I put improvements in quotes because it's with respect to commercial purposes, where you really want a healthy, clean E. coli, which brings up this question of genetic node. So the rest of it is just showing you all the different things that we have observed by a more or less unstructured approach. These are advantages that can be used in practice, but they're basically phenomena that come from finding things that are from the part of the genome or the contaminating genomes that are basically trying to destroy the host and getting rid of them. And I also want to thank these people. So I appreciate your letting me go a little bit over time, but I definitely wanted to give Click and Clack their due.